Hi, good afternoon, class. Today I'm starting a new chapter on replication, and this is the sixth chapter in the book. And on the syllabus, it is chapter number 3rd. So, first chapter we talked about the history, the second chapter about the chromosomes, and this is the third chapter and on the syllabus and sixth chapter in the book. And it is from in fourth edition of Essential Biology. This is from pages 197 to 222. Again, a few advices. We are almost advancing on time with the syllabus and going from chapter to chapter. You are doing pretty good job with the assignments. Just few suggestions. Still go back and review the syllabus if you have not. Listen to the recording, take notes, submit the assignments on time, and so on and so forth. And also pay attention to my office hours, which are given on the front page of the syllabus. So let's start. What's the objective of this chapter? The objective is to understand the process of replication. Why is replication important? When does replication occurs, right? Mechanism of replication, proteins that mediate replication, errors during replication, how is DNA damaged? These are the important topics or objectives that we shall be covering, and I would try my best to explain these events which happen within the cells. So just briefly, why is replication important? So replication is important because any living organism has two essential properties. That's why they're living. And one is growth, and second is development, and third is differentiation. This you may not be seeing these three in bacteria, but of course, bacteria you will see the growth. In bacteria, the growth is nutritional dependent. The moment there's a food nutrition in the media, in the environment, bacteria will thrive, and after that it will look for new food. In case of multicellular organisms, it could be just division of cells, or just say, let's say in case of humans, there's not only development, division, but there's a differentiation of the cells, tissue, and there's a hierarchy in our body in terms of humans. Fourth very important thing is repair. Right? Our body in humans or multicellular organisms, it's constantly under wear and tear. For example, we lose intestinal lining once in a while, and then it has to be replaced by new intestinal lining. Secondly, our skin, we shed skin. It is a normal processes, and so new cells have to come in to replace the old ones and those who have gone out. Or let's say there is a cut in our finger. So where does this new skin comes from? This new skin comes from cell division. But cell division is what? Cell division is a process by which one cell is divided into two cells. But what is more fundamental to cell division is the chromosomal duplication. And most fundamental to chromosomal duplication is that DNA replication. So before cell cycle occurs, right, the DNA has to replicate. After DNA replication, chromosomes have to duplicate, right? Chromosomes have to duplicate. And after chromosomes have duplicated and the chromosomes have divided into daughter chromosomes, divided, right? Then new cells have to be formed. So if the replication is good, chromosomal duplication will be good, chromosomes will divide and two new daughter healthy cells will be formed. If replication is wrong, then everything becomes wrong. And then what happens that then if the replication is not right, then it can give rise to diseases like cancers, which are very difficult disease. So that's the significance of replication. When does replication occurs? Replication occurs when cells are poised to divide. There is no point replication to occur if it is not needed. So the moment there is a wound, right, then you will see first blood clotting, or new skin comes out. So it only comes out when it is needed, right? Then mechanism of replication. So replication means it occurs in phases. It is not haphazard. It moves from steps to steps. There are players which work as a team to help each other. It's not a single-handed process. It is done by a battery of proteins, a team of proteins that work towards duplicating their DNA. It is very, very delicate process, right? Because a mutation gets in, it can give rise to very severe diseases. So there has to be a mechanistic control of this process. And these players are what? These players are mainly proteins and, of course, nucleotide, right? When the daughter DNA is being formed, new DNA is being formed, you need the building blocks, the nucleotide. So proteins are the major players, and nucleotide also are the cofactors, right? Substrate. And so the length of DNA is few meters in our body, so there is bound to have mutations or errors. And nature has developed mechanism to get rid of those errors, to rectify those errors, but it may not be rectified and it could be lethal, right? So for example, you're writing a term paper, you're writing a paragraph, right? You write a sentence or a word. You look back and you check at the spelling, typos, right? So what is that? Proofreading. So these enzymes, these proteins also have proofreading ability to correct the mutation while in the process of replication. Remember, when replication is occurring, few meters of DNA is getting duplicated, right? And then also our, look at the environment. Our body is constantly bombarded with various mutagens or type of radiations which would damage the DNA. So to understand or to develop treatment for diseases like cancer or wound healing or some problem with DNA damage response, we have to understand the replication, right? So, so look at, pay attention to this slide. So our first point that I would say in this slide is that there is a hierarchy in our body. Cells, right, fundamental building block of the life. You have cell theory from chapter 1. Then these cells, identical cells, they come together, they group together to form tissues, and then large different kinds of tissues, they come together to form an organs, and organ forms systems. So in a particular day, at least 300 billion new cells are formed, which means that, as I mentioned to you, formation of new cells is dependent upon DNA replication, which would translate into 200 million per minute cells. So our body, even we experience or not, goes through so much of replenishment of replacement of old cells with new cells, micro-injury, RBC, they are removed 3 to 6 months, that our body is continuously manufacturing new cells to do the work. And if there is any deficiency in this process of replication or daughter cell division, then there are severe consequences. So replication is important for development also. Why? Because as a human being, we start our life as a single cell zygote that divides, that develops in 9 months into an individual. And the essence of this is cell division. If the timely cell division doesn't occur in a zygote, it could be a potential problem, developmental delay or other kind of disorders, right? So number one, it's cell replication or DNA replication is important to maintain our body physiology by continuously dividing. Then it is important for development, right? And if there is any problem in replication, it can be passed on to next generation, and it could give rise to severe diseases. And also I mentioned to you of cancer. Any abrupt division mutation can trigger a cascade of events that can lead to tumor development and metastasis, which could be very difficult, right? So watch this video on replication. And now once we studied about replication, we need to know where does replication occur. As I mentioned to you that cell division is a very planned process. It is not haphazard. So it occurs in our body as a process called as cell cycle. And this cell cycle is composed of G1 phase, which is called as growth phase. This growth phase is followed by S phase, which is called as synthesis phase. This is the synthesis phase in which DNA replication occurs. And then cells enter G2 phase, then mitosis, where within mitosis you have prophase, metaphase, anaphase, telophase, cytokinesis, and 2 new daughter cells are formed, right? So S phase is the phase where replication occurs. So, and you know from chapter 1 that replication is a semi-conservative process, and it was discovered by Messelson and Stahl, right, using the radioactive nitrogen 15. So let's say if this is the parent DNA, if this is the parent DNA, right, and this parent DNA has to duplicate because it is necessary, because there is some need or there is a wound, so DNA needs to duplicate or replicate. So this is mediated by a battery of proteins. So what happens that if DNA has to duplicate, DNA has to unwind, right? So you have to consider DNA like a zipper. So if it has to unwind, the 2 strands have to open up, right? And after the 2 strands, they open up, then the new daughter cells have to be formed, right? And this new daughter cells formation has to be perfect, right? If it is not perfect, then it could be a problem. So slowly, the whole genome, the whole DNA gets duplicated, and ultimately, what is the end result? We get 2 daughter DNA, which are complementary to each other, and this DNA, which is a source of genetic information, is passed on to the daughter cells. Right? So the ability of a cell to proliferate, right, in a coetic depends upon accurate duplication. So DNA is there and it is replicated. There could be stress on the DNA because of mutagens or environment, and DNA could mutate. In addition, DNA also changes, and that is called as variation. So look at this basic, very fundamental slide, right? So this is a parent DNA, right? This is a parent DNA. And these are the daughter DNAs. This is the parent DNA. These are the daughter DNA. This is the parent DNA, right? So see, please note here that these double-stranded DNA, which are connected by nitrogenous bases through hydrogen bond, and one of the strand goes from 5' to 3', and other strand goes from 5 to 3. So what I want to say that DNA is anti-parallel. Anti-parallel means they are parallel, but their orientation is opposite. So let me explain to you here. So what I mean here by anti-parallel? the DNA is two-stranded, right? So these are parallel. These are parallel. But why it is called as anti-parallel? These are called anti-parallel because one of the strand is from 5 prime, the other strand is 3 prime, right? Similarly, here also, the 5 prime strands would be 3 prime, and the 3 prime would be. So this this makes DNA as an anti-parallel molecule. They are parallel, but their orientation is opposite. So anti-parallel, okay? That's the meaning of anti-parallel. So this is a parent DNA going from 5 prime to 3 prime, 3 prime to 5 prime. And then both these, so these are, orangish or yellowish color, the two strands. These two strands, they separate out, they unwind and give rise to two new daughter strands, right? So this is a semi-conservative, half old, half new. Half is conserved and half is new. So that's the mode of replication, right? So if you see here, the DNA is unwinding and the daughter strands are used as a template to synthesize the daughter DNA, right? Now, I'm introducing to you two terminologies, right? Two terminologies, right? One terminology is called as leading strand, right? And the other terminology is called as lagging strand, right? So remember these two terms, leading and lagging strand, right? Leading strand is also called as continuous strand, and the lagging strand is also called as discontinuous strand. Leading versus lagging, continuous versus discontinuous. And remember one term in addition. Discontinuous is associated with another term called as Okazaki fragment. So what did we learn here? That if the DNA has to serve as a template for the synthesis of daughter strand, the DNA has to unwind. The double-stranded has to become two single strands. And then each single strand serve as a template, right? So one of the template is continuous. The newly synthesized strand is continuous. The other strand is discontinuous. And discontinuous means the DNA is synthesized in fragments. And these fragments are called as Okazaki fragments. So I will come back to this, but remember this terminology is going forward. As I mentioned to you, that replication is not haphazard. It happens in steps, right? So what are the three major steps? Initiation, right? Initiation, and then elongation and termination. So why these phases? These phases is to control the randomness, to make the process seamless, to control the process. So first, just like, when we start a cycle or a bicycle or a motor car, you have to turn on the engine and then you have to press the gas. And then when you reach your destination, you have to stop your car. Now, why do you stop your car? You stop your car because to save the gas. If you lose the gas, you lose your dollar amount. So you have to conserve, right? So that way, by same perspective, our cells are also economical. They do things in phases. Get the things started, get the things moving, and when the job is done, it has to stop. Initiation, elongation, and termination, right? Now, as we are moving, I'm going into the details, right? Step by step. So what happens so this is so in the top panel, what you see, So this will be the top panel. What do you see here, right? You see here that the DNA is double helix, anti-parallel, each strand running from 5 to 3, from the top strand running from 5 to 3 and the bottom strand running from 3 to 5. Now, if this DNA is set for replication, right, then in the middle of this DNA, there will be a generation of a bubble. This is called as a replication I, right? How is this formed? This bubble is formed because hydrogen bonds between the double strand has broken. It has broken to facilitate the synthesis of daughter strands, right? So this is called as replication I, or origin of replication, so on and so forth. Now I want you to focus on two things here. I want you to focus when the DNA is unzipping, right? When the DNA is unzipping, it is, if you see carefully, right, it gives rise to a structure like this, right? Here, structure like this. Right? Similarly, here, also a structure like this. Right? And these structure, if you look at it, they look like the alphabet Y. They look like alphabet Y. Right? So they look like, as I mentioned to you, alphabet like Y. Right? So let's say this look like structure like Y, right? Here, Y. Similarly, I copy paste and change the orientation. This also looks like something like Y. These points, these 2 points, they are called as replication fork, right? These are the two places where the DNA is beginning to unzip and it is going to start replication. So this is called as replication I. Replication I. Okay. So let's talk about key concept. These replication I, or replication bubble, or origin of replication is formed by breaking of hydrogen bond. And in bacteria, this origin of replication is around 100 nucleotides. And bacterial genome is circular, and there is only one replication of origin in bacteria. In human genome, which is meters long, the human genome has around 10,000 and overall, 220 chromosomes. So each replication has 2/4. So, there is, in the bottom message is, in bacterial chromosome, there is only one replication I, right? But in human genome, which is meters long, there are hundreds of replication I, or bubble, that is going on in parallel. And what I'm showing to you in next slide, you will see that. So this is a human genome, right? Human genome, or let's say human DNA, which is very large, right? So you see here, there are multiple fork of replication here, right? There are multiple fork, but in bacteria, it is only one. Okay, so keep this in mind. Now, this is the most important, and I want you to focus here. Right? what I want you to focus here is on this bubble, right? Focus on this bubble or fork. So I'm focusing on this one. So the replication the replication always occurs in 5' to 3' direction, right? So what I mean here, let me show you that, let's say these are the two strands which have become separated, right? So this is 5' and this is because anti-parallel, this is 3', right? Similarly, this one is 3', this one is 5'. Now, it is not known what is a selective mechanism, but either one of this DNA could serve as a template for replication. And replication always occurs from 5' to 3'. The direction of replication is from 5' to 3'. Now, I will go into details later, why 5' to, but remember, in deoxyribose sugar, the 3' carbon atom, the OH group, is most suitable for chain elongation. So a cell can choose this strand for duplication or this strand for duplication, whatever it chooses, right? So if, let's say, if the replication is chosen on this strand, right? So replication will always occur from 5' to 3'. So from this direction to this direction, replication will occur, right? Now in the opposite strand, it cannot occur in same way, right? In opposite strand, the replication will occur on bits and pieces in a fragment, right? Like a fragment, then this fragment. So this strand, which is chosen from 5' to 3', this becomes a continuous strand, and this is a leading strand. But in the other strand, where it is slow a little bit, it becomes a lagging strand, okay? So leading versus lagging strand depends upon which strand is chosen to serve as a primary template, and the replication occurs from 5' to 3'. So let's go back to that slide, right? So look, there are two strands here, DNA. One is going from 3 to 5, another is going from 5 to 3, right? So if it has to go from 3 to 5, then 5 to 3 is the continuous strand, right? And the other strand becomes the lagging strand, and it synthesizes the DNA in fragment, and that is called as lagging strand with Okazaki fragment. Now, in one eye, right, in one replication eye, there are two forks. Let me explain again. In replication eye, there are two forks, two replication fork. What do I mean by this? That this is fork number 1, right? This is fork number 1, and this is fork number. So in one replication eye, there are two replication fork, 1 and 2. Okay. So when the DNA is poised to replicate, the two strand has to separate, right? How does it separate? Because the hydrogen bonds are unzipped, right? So how a DNA is unzipped? So this, I'm going into more detail. DNA is unzipped by a protein that is called as helicase. I'll come to that later. So this is the true template, and it is unzipped, what you see here, by an enzyme called as helicase here, this point. So unzipping means the two DNA strands, they are separating. So this is the point where the enzyme helicase is unzipping. And then the strand that goes from 5' to 3', it is called as leading strand, and the other strand automatically becomes a lagging strand, and there the fragment is synthesized in bits and pieces, or fragments is the right word, that is called as Okazaki fragments. Okay, now let's look at more convincing picture. So DNA is unwound by an enzyme called as helicase, right? Helicase is the enzyme that unwinds the DNA. That unwinds the DNA, right? Now, when the DNA is unwound, one of the strands will serve as a leading strand. The leading strand, the DNA synthesis will occur in 5' to 3'. In the lagging strand also, it will occur in 5' to 3', but it will occur in fragments called as Okazaki fragments, right? So a leading strand, lagging strand, Okazaki fragment, and enzyme helicase. Now, one thing I would add, another player, is that when replication is occurring, right? When replication is occurring and the daughter DNA is being synthesized, right? So when daughter is being synthesized, one thing is very important, that the enzyme that carries the process of replication is called as DNA polymerase. So this is the main actor of DNA replication, right? DNA polymerase. DNA polymerase is in the enzyme that helps in the excuse me, in the synthesis of daughter strands. Then the DNA is unwound by an enzyme called as helicases or helicase. Now, one thing, DNA polymerase is very powerful enzyme, right? But it cannot synthesize the DNA by itself. It needs a help. It needs the help initially. So what is that help? Is that, let me share with you. So let's say this is the one strand, right? And this is the another strand. For sake of example, let's say that this is the continuous strand, right? This is a continuous strand going. But the point I'm making, that DNA polymerase cannot start replication on its own. It needs a help. And what is that help? What I mean by that help is that it needs a pre-existing RNA, right? It needs a pre-existing RNA. This RNA is called as RNA primer, and this is synthesized by an enzyme called as RNA primase, right? Some books will also call this as DNA primase, but the ultimate thing is that the message I'm conveying to you is that the DNA polymerase is the major enzyme that helps in the process of replication. but it cannot start on its own. It needs help of pre-existing RNA. And that RNA is synthesized by RNA primase. So let's add one more player to this. Here, this process of replication is primase. So DNA polymerase, helicase, primase, right? 3 enzymes. And of course, there is leading strand and lagging strand. So let's go back to the slide. Right? So this is topoisomerase, is an enzyme that, because our DNA is super packaged, so it has to be unknotted, right? And that unknotted, that DNA is straightened, that knots are removed by topoisomerase 1, topoisomerase 2, DNA gyrase that we will talk later. So in addition to primase, helicase, and DNA polymerase, you have topoisomerase 1, topoisomerase 2 that helps in removing the knots of DNA to straightening the DNA for replication to occur, and also DNA gyrase, right? So leading fragment and the lagging fragment, it needs RNA, and this RNA is being synthesized by RNA primase, so on and so forth, right? So now let's again focus on this slide. Here I introduce to you what some books, they say let me caution you here. Some books will say the primer that is synthesized, which is RNA, by RNA primase. Some books will say DNA primase. So from here on the slide, because it says DNA primase, so we'll take it as DNA primase is the enzyme that synthesizes pre-existing RNA to help DNA polymerase to start the process of replication. Then you have DNA helicase that unzips the gene, the DNA. You have DNA topoisomerase 1 and 2 that helps to unknot the DNA. That is DNA gyrase. And then of course you have DNA polymerase that helps to synthesize DNA in which direction? 5' to 3'. So let's look at the list of all the players that are involved in the process of replication. DNA helicase, DNA polymerase, DNA clamp. It is a protein which prevents DNA polymerase from dissociating. It's like a clamp. Now the most very important protein that I want to mention in this slide is single strand DNA binding protein. Okay, so what happens is that when the DNA is separating, right? So, so let's say this is the DNA, right? From here, and what I'm trying to show it to you is that, let's say DNA is separating here, right? It is separating. This is the replication fork, right? This is a replication fork, which is DNA is unzipped by DNA polymerase. Now, when the DNA is single-stranded, right? So when the DNA is single-stranded, it is wobbly. It is moving, right? It is moving. So there's a protein that tries to stabilize the single-stranded DNA strand. This protein is called as single-stranded binding protein. It only binds to the single-stranded DNA. That's why it is called as single-strand binding protein. Single-strand binding protein. So this single-strand binding protein, this binds to the single-stranded DNA and tries to stabilize the DNA such that DNA polymerase can slide through the nucleotides of the single-strand DNA and helps in the synthesis of daughter strand. Okay, so let's go back to the slide. So then topoisomerase, it relaxes the DNA, the supercoiled nature. Then DNA gyrase relieves the unwinding DNA by helicase. So read this, okay? Then DNA ligase, right? DNA ligase is the enzyme that seals the Okazaki fragment in the end. Okay, so Okazaki fragments are formed. So let's go back to the board. So Okazaki fragments are formed due to discontinuous, right? So this is let's consider these as Okazaki fragment, right? Okazaki fragments. So the last nucleotide on the Okazaki fragment is sealed by an enzyme called as DNA ligase, right? DNA ligase is the enzyme that seals two nucleotides in the end and then make the strand continuous, right? So in the lagging strand, right? Lagging strands, right? Lagging strand has Okazaki fragments, and this in the end is sealed by an enzyme called as DNA ligase. So there are many players, as I mentioned to you, during the process of replication, and it's the most important process. Okay. Then we'll look at, so within primase provides a starting point for RNA for DNA polymerase to begin synthesis of new strand. Primer is 10 nucleotides long, right? Synthesized every 100 nucleotides, right? Then eukaryotic we'll talk about telomerase. So here I have put a video that you must watch to review the process of replication. So this is actually the backbone of DNA, right? The single strand. And this single strand, it serves as a template, right? The single strand serves as a template for the synthesis of DNA. Right? So this is the single strand DNA. Right? This serves as a template. So if it is G guanine, then the base that will come on the complementary strand, new daughter strand is C. If it is G on the parent template, then the daughter template will have A, G and C. So what is this called as? Complementarity, right? And this chain will grow. Okay. But what is unfortunate part is that sometimes when the chain grows, right? When the chain grows, there is high chances of problem, high chances of incorporation of wrong bases. It can happen because DNA polymerase has to incorporate millions of bases, and it could go wrong. It could be because of many things, right? Our cells are under continuous stress. So proofreading comes into play. So these DNA polymerase have multiple abilities, multiple activities, right? And one of the activities is called as proofreading, right? So when you write a term paper, when you write a sentence, when you write a word, it can go wrong, right? Intention may not be wrong, but it could be wrong. So you can go back and review. That reviewing process is called as proofreading. So these DNA polymerase enzymes, they have the property to go back and correct the bases till the time it has time to do it. If it is late, then it can't help, right? Then the mutation is passed on and from one cell to the next cell, and then it can give rise to many diseases, right? So proofreading. So self-correcting. So all these, so there are 3 DNA polymerases in bacteria that we will see later. All these 3 DNA, they not only have the ability to synthesize daughter strand, but also have ability to proofread, to remove the wrong bases, right? So what happens that, so let's focus. So let's focus on panel number 1, right? What do you see here? You see here that the DNA polymerase is synthesizing. And what do I mean by synthesizing? There is addition of complementary bases to the template strand. And then by mistake, there is incorporation of wrong base. So what you see here is incorporation of wrong base, right? And then there is a problem. So how to fix this? So this is fixed by proofreading. So what happens that when DNA polymerase 1, it is advancing, or DNA polymerase 3, it is advancing to synthesize daughter strand, putting complementary base, and there is an error, they always look back and try to fix it, right? So when they look back, the orientation is changed. So the polymerase activity is from 5 prime to 3 prime, and the proofreading activity is opposite, 3 prime to 5 prime, right? And then it looks back, corrects the base, and then again moves ahead. It removes the activity, the wrong base, and that activity is called as exonuclease activity. Right? So this is a proofreading. So this enzyme, you can think like a hand, and the DNA is moving from one end and coming out from the other end, and it has the ability to proofread. So DNA polymerase has two abilities. One is polymerizing and the second is proofreading. Okay? So let's look more closely here. So look at panel number 1, right? So, so see here, the orange strand is the parent strand. In the blue color, slide blue color, it's RNA primer. It serves as a entry point for synthesis of discontinuous strand or lagging strand, and that is Okazaki fragment. And slowly, this Okazaki fragment has to be filled in, right? So to be filled in, the RNA has to be removed, right? DNA is not a chimeric molecule made up of RNA and DNA. It is totally DNA. But for replication, it does need RNA primer. And that RNA primer has to be removed. Again, that is removed by DNA polymerase 1, right? So DNA polymerase, so which enzyme removes the RNA from Okazaki fragments? The DNA polymerase 1 through its proofreading ability. What is that proofreading activity? I would say 5 prime to 3 prime. So let's talk here a little bit. DNA polymerase 1 has 3 activities. Number 1, polymerizing activity 5 prime to 3 prime. Proofreading activity from 3 prime to 5 prime exonuclease. Then removing the RNA primer, that is 5 prime to 3 prime. So look at this, look at the heading of this slide, right? What does the heading of the slide says? It says that removal of RNA primers and joining of Okazaki fragment. Because of its 5 prime to 3 prime exonuclease activity, DNA polymerase 1 removes RNA primers and fill in the gaps of the Okazaki fragment. Right? So a lot of information, listen carefully and try to take notes, right? Now, let's focus on telomeres. What are telomeres? Telomeres are the sequence of DNA or chromatin at the end of the chromatids, right? So at the end of the chromatids. Right? Now, what is it made up of? Right? Telomere is highly rich in guanine and cytosine. Right? So try to understand the significance of guanine and cytosine. Guanine and cytosine are connected by triple bond as compared to adenine and thymine. Guanine, the triple bonding makes guanine and cytosine more stronger, difficult to break as compared to adenine and thymine. This concept will help you also. So telomeres are at the end of the chromosomes, right? And they are mainly rich in guanine and cytosine, right? So telomerase is the enzyme that helps in the replication of telomeres, right? So look at this video on telomerase function, and I come back here to describe you the mechanism. So telomerase is an enzyme, right? But it's very interesting, right? Telomerase is an RNA protein complex, meaning that telomerase has a protein part that helps in polymerization of chromosomal end, but it also has an RNA. Now, why does it has RNA? It has RNA because remember that RNA is needed to kickstart the replication. DNA polymerase is dependent upon RNA. So this RNA, which is part of telomerase, helps in kickstarting the process of telomeric replication, right? So this is a very interesting table that mutation can occur. So we are writing an address to pay our bills or sending a card. We can exchange the zip code, and then if the code is exchanged, it's similar to mutation, right? And then the letter will never reach its destination, no matter your intention is very good. So error happens, right? So that's the analogy I can give. So this could be a chemical or non-chemical processor. So depurination of guanine, it can create new kind of base, right? And which can confuse the DNA polymerase, leading to synthesis of wrong DNA strand. And that can be passed from one generation to the next, right? So we will read a mutation in more detail in few chapters later. Similarly, look at deamination. Deamination means removal of amino group, right? Removal of amino group. What is amino group? NH2, right? So look here. If NH2 is removed, right, then it becomes cytosine, deamination, right? So cytosine, amine group is removed, it becomes uracil, right? And uracil is a hallmark of RNA. So cytosine is supposed to bind to or form triple hydrogen bond to guanine. But if deamination of cytosine occurs, then cytosine becomes uracil. Uracil will have more affinity for adenine as compared to guanine. What happens? The GC now changes to AU, then AT, and so on and so forth. So it's a big change if it happens into a crucial protein like hemoglobin or P53 or any cell cycle kinase. So look here, ultraviolet rays, if there is a sequence of DNA in which there are two consecutive thymine, right? So when ultraviolet light falls, it can give rise to thymine dimer. So two thymines, they come and they bound covalently. So look here, they bound covalently, right? And this is called as thymine dimer. That's a problem because thymine dimer is a covalent bond. And that can impede the rate of replication. And then if replication is delayed, then daughter cells could be abnormal. So chemical changes in these nucleotides can have bigger consequences on a cell. And it can lead to very severe diseases. And there's a chapter on cell cycle where we will study how a normal cell becomes cancer because of mutations. So read this chapter slide, it gives an example that how a base is incorporated and then it mismatches A with G, C with T, vice versa. And then the whole generation is changed. These are I have posted some of the important slides. So this is the difference between prokaryotes and eukaryotes of replication, right? So the primer length is 50 nucleotides in RNA, in bacteria. Bacterial polymerases are 1, 2, and 3. Number of origins is only 1. The length of oligonucleotide in bacteria is 100,000 to 2,000 nucleotides. Rate of replication, 500. So bacteria divides faster. As compared to humans or eukaryotes, RNA primer, 9 nucleotides. DNA polymerases, alpha, beta, gamma, delta, eta. Number of origins, replication origins, multiple. How many nucleotides in Okazaki? 200. And then rate of replication is just 50. This slide I got from a site on the internet. It shows differences between DNA polymerase 1, 2, and 3. Now focus here on the proofreading. DNA polymerase 1. It has 3,5 exonuclease and 5,3 exonuclease, right? And all these DNA polymerase 2 and 3, they have only 3' to 5' exonuclease activity, right? So these tables are very important from exam point of view and for your knowledge, because when you are reading about DNA polymerase 1, 2, 3, or alpha, beta, gamma, delta, eta, we get lost. It is nothing unusual. But when you put your information in the form of table or in form of chart, it is more you're able to retain. So again, these tables are very important. Okay? In this table, there's a difference between DNA polymerase alpha, delta, and eta, gamma of eukaryotes. Right? Look at the direction of their polymerase activity, 5' to 3'. The delta has polymerase activity and exonuclease proofreading. Eta, they don't have proofreading. Gamma, it is mainly mitochondrial DNA polymerase. So a lot of information is here, and everything is important from your exam point of view, right? And in the end, you again review the details of bacterial and eukaryotic DNA polymerase. It's very, very important. So we are now getting into the woods of replication and molecular biology, and this chapter is very important. Okay. So thank you for your attention and have a good time.